Hi, everyone. Thanks so much for coming to this session today. I'm Melanie Cree. I'm a pediatric endocrinologist, and it's my great pleasure to introduce my longtime colleague back to 1995 and friend, Dr. Cecilia Diniz Behn. She's a candidate for faculty position within the Department of Bioinformatics and for the chair of research for the Ludwig Center for Women's Health. She started her mathematical and women's health focused career at Bryn Mawr College, which is where we met. And she received her master's from the University of Texas and then her PhD in mathematics at Boston University. She then did postdoctoral fellowships at Harvard and the University of Michigan. She's now an associate professor in mathematics at the Colorado School of the Mines and is a adjunct faculty at CU Boulder and also an adjoint faculty in our division of pediatric endocrinology. And she's had that position for about 8 years now. Her research is at the interface of mathematical modeling, data science, physiology, and she focuses on scientific and translational questions in sleep-wake behavior. That's her personal research interest in circadian rhythms. And then she's delved into the world of whole body glucose metabolism and integrative physiology and really helped us with understanding that in our division. Her research and professional activities really prioritize women's health and equity issues. I've attended her classes at Mines, and they're 70% female for her mathematics classes. It's great. And we have many publications and grants together. She's a fantastic all-around academic faculty, and her presentation today, entitled Mathematical Modeling of Metabolic Dynamics in Adolescent Girls, is showing some of our work. So I will let her take the stage.
Great. Well, thank you so much, Melanie, and thanks to all of you for the opportunity to be here and tell you about some of this exciting work that we've been doing and how I think it can really move forward in this position generally. So I think you heard a little bit about this, but my research interests really lie sort of at this interface of sleep, circadian rhythms, and metabolism, and particularly in the interactions among all of these systems. And the lens that I use to look at all of this is mathematical modeling. A lot of data science and analysis as well coming into this. And a lot of the questions that I look at are really focused on questions in women's health that are particularly understudied in this space. So a quick overview of what I'll be talking about today. I'm really going to be focusing more on the metabolic side of the work today, although I'll tell you a little bit about some associations with sleep and circadian factors. But I'm very interested to talk to people who are interested in sleep and circadian questions as well. So, please ask about that in the questions or follow up with me afterwards. I love to talk about that work as well. It's just always a matter of time. So I'll be starting off with a little bit of background dynamics. I know that we have quite a diverse audience, and I just want to make sure that we're all on the same page. I know we have some true experts in this area as well, so I'm just going to be talking about some of the basics, but just so that you can kind of understand what we want to do with the modeling. And then I'll talk about 3 different projects that kind of highlight some of the capabilities of my group in using mathematical modeling to quantify different aspects of metabolism with a focus on adolescent girls. And those are projects all with faculty who are at Anschutz now. And then, as I said, I'll touch on this project looking at some interactions between sleep and circadian factors with some of these metabolic factors. And I'll just touch a little bit on my vision and future work. I know there's an opportunity to delve into that more tomorrow, but I just wanted to give you sort of a sense for where my work is heading. So I just want to make sure I know what time it is, so I can be a little bit aware of that. I don't think it works, but 11:08. 11:08, thanks. Okay. So one of the key reasons for looking at adolescence is because puberty is really associated with some significant differences in metabolism, so reduced insulin sensitivity, insulin hypersecretion, and then also on the sleep and circadian side, there's a later circadian timing, so this sort of circadian phase delay that you may have heard about that's sort of leading to this movement to have later school start times for high school students. To improve their health and their capability of functioning a little bit later in the day. And obesity and impaired glucose tolerance is an epidemic at all life stages, but this is particularly growing in the adolescent population and currently affects over a third of US adolescents. And the prevalence of type 2 diabetes also is increasing in youth and presents much more aggressively. And so there's really a need for therapeutics. Many times adolescents don't respond to therapeutics as well as adults, and the time course of the disease is much faster. And then thinking about girls, in addition, in adolescence, there's a prevalence of type 2 diabetes that's higher in girls than in boys. And the sort of reason for this pathogenesis of disease is not understood at this point. This is one of the things that we're trying to understand with some of this research. But, in adult men and women, the incidence is more similar, but at adolescents, it affects girls more. And another reason that we're looking at girls, so in my work with Dr. Cree, we've been doing a lot of work looking at specifics of metabolic dysfunction in girls with polycystic ovary syndrome. And so with that, then we're often considering obese controls, also are girls. So when we think about metabolic dynamics on both the clinical side and the research side, a lot of measures that focus on this really look at steady state behavior. So clinically, this might be something like the HOMA-IR, which is a quick way to quantify insulin sensitivity based on fasting glucose and insulin levels. On the research side, on sort of the other end of the spectrum, we have very involved techniques like the hyperinsulinemic euglycemic clamp that keeps glucose at steady state under different conditions, elevated insulin, and allows the very detailed assessment of glycemic control. And in the case of the multi-phase clamp, really enables the researcher to look at tissue-specific insulin resistance. And then for insulin secretion, hyperglycemic clamps can be used to look at that. And so there's a lot of information that you can get from these techniques. And so they're very valuable, but metabolism is really a dynamic process. And it's something that's changing across the day and as you eat and drink. And so really, if you take into consideration the dynamics of these things, there's a lot more information that you can get. It's a lot richer data. So that's something that we really wanted to focus on here. So just quickly to remind everybody, in terms of maintaining a constant source of glucose for your brain and body function, we have different places that we can get it from our food, from the liver, from fat, and the source really changes depending on what physiological state you're in. So if you haven't eaten in a while, you're in a fasted state, then the liver is the primary source of glucose. That there can also be glucose that's derived from adipose tissue that's broken down. And when you're in the fit state, food is your primary source of glucose, but typically the intake that you have is more than you really need at steady state at that moment. And so you have extra glucose that has to then be taken up and stored, as well as the suppression of the glucose that you were previously getting from your liver and then derived from your adipose tissue. And so insulin is really the star player here in driving these really rather dramatic transitions in what's happening in the function of these different systems. So it's released from these pancreatic beta cells, and it acts in multiple ways to affect both the disposal of glucose, so the uptake from muscle, fat, and liver cells, and the production of glucose by suppressing this glucose that's coming from the liver and the adipose tissue. And insulin resistance occurs when the normal response to insulin is blunted, and so you need more insulin in order to have the same level of control. So we know that excess weight and physical inactivity can be big contributors to this, and insulin resistance can also eventually contribute to beta cell failure. And so then you have less insulin around, and then sort of a vicious cycle is created. And as I mentioned, insulin resistance may be tissue specific, and so the response of individual tissues could be preferentially or differentially impacted. And so this could affect the glucose uptake, the glucose production, or lipolysis. And so really getting a good understanding of what's happening with tissue-specific IR can really help in driving personalized medicine and thinking about signatures of different kinds of diseases and the metabolic dysfunction that's associated with them. So when I talk about dynamics, what am I really referring to? So this is just an example from a healthy adult undergoing an oral glucose tolerance test. And so this is a person who would come in fasted. And so we can see this sort of steady state level where they're starting off. And then at time 0, they're ingesting a sugary drink that's causing the glucose concentration to spike up. Then the insulin concentration responds and is released. And then that brings glucose back to baseline, and then insulin returns to baseline. So that's what we would sort of generally expect as a profile in a typical healthy individual. So when we think about the projects that I want to talk about today, we'll focus first on this idea of glucose insulin dynamics that I was just talking about, and how we wanted to assess full body insulin sensitivity in girls with polycystic ovary syndrome. Then I'll talk about a novel model that we developed for glycerol insulin dynamics. And then finally, I'll talk a bit about C-peptide dynamics and using that to assess beta cell functions. In that case, that will be with adolescents with cystic fibrosis. So this was work with my collaborator, Melanie Cree, and with my PhD student, Kai Bartlett, who's now graduated. And so here, polycystic ovary syndrome is so sort of surprisingly prevalent disease with a huge economic burden, and is associated with or as a leading cause of infertility, but also has a strong metabolic profile. And so understanding sort of where these higher rates of insulin resistance, fatty liver disease, cardiovascular disease are coming from in this population is a big focus of my collaboration with Melanie. And so she's established previously using clamp techniques that there is insulin resistance in muscle, liver, and adipose tissue in girls with PCOS. And really a long-term goal of our work is to understand the pathogenesis of this and be able to identify effective early state interventions. So the motivation for this study was to be able to understand what's happening in this dynamic context of an OGTT. We can't consider various hormones that are involved in the gut if we use a clamp, because things aren't going through the gut. So we wanted something that was more physiologic. And in terms of sensitive measures for estimating insulin sensitivity from oral glucose tolerance test data, there's an oral minimal model that's been developed by Delaman and Covelli and colleagues. And so we wanted to apply this model, but we found that this model needed a lot of adaptation to actually be applied in our population. So this just shows you the data, so you can see sort of generally what this looks like. This is a research frequently sampled OGTT. So these are the sampling time points across the top. And then you can see the average glucose and insulin response kind of looks like what we saw in the previous example, although you'll notice that the time course is much stretched out so that people aren't coming back to baseline, usually for over 4 hours. And there's also a lot of variability, including some very high insulin insulin release, which again can reflect what's going on with the adolescents here. So some of the particular challenges with the data set we were working with were these high insulin concentrations. And it's known that the oral minimal model can give spurious results when insulin concentrations are very high. And also the fact that the model works best when you have the full-time course of data, have it returning back to baseline. But in our participants, that needed 6 hours. And so this represents a great participant burden because people have to fast and then come in and have this sugary drink and then wait for 6 hours to come back to baseline. It's also very important to think about clinically because people usually are not going for 6 hours between meals. And so that means that this population is not really returning to baseline between meals, which has other implications for sort of how their actual daily glucose insulin dynamics are playing out. So in terms of the approaches that we thought about for this, there was some previous work that had demonstrated that assuming some exponential decay in what's happening with the rate of appearance after a certain time could be helpful. And so we developed a collection of models that both considered different durations of data and did or did not assume an exponential decay after the data that we were assuming, and then compared our resulting estimates of insulin sensitivity. So here's a quick schematic that gives you an overview of the model. And so I wasn't sure the balance of equations and not equations to show in this talk because we do have such a diverse audience. So I will show the equations, but sort of the intuition for the equations you can just get from this schematic. So all of the models that we'll be working with today are compartment-based models that are really assuming this sort of general compartment of, say all of the glucose in the blood is what we're measuring here. So here we're thinking about the sources of glucose as that endogenous glucose production that's coming from the liver and adipose tissue, and then the drink that's been ingested. And then there's uptake of glucose that's both insulin dependent and insulin independent. And the typical way in these models that insulin is handled is to think about insulin concentration as driving the source of insulin action. And this insulin action term, I would say, is a little bit hand-wavy, but it essentially captures some of the complex dynamics that happen as you go from the measured insulin concentration to its functional effect physiologically. So here are the equations. And so just quickly, here you can see within the glucose equation that we have this production of glucose that's primarily driven by this rate of appearance of the glucose coming from the meal. And this is something that we typically just estimate a piecewise linear function to represent. And then we have the uptake of glucose that's both insulin independent here and then insulin dependent here through this X term, which represents insulin action. And insulin action is just something that decays when insulin is below basal levels and then grows according to how much insulin is above basal levels, where IB is representing the basal level of insulin. And so then typically from this model, this ratio of parameters P3 over P2 times the volume of distribution is what is taken as the measure of insulin sensitivity. And so what we did, as I said, we generated this collection of different models and then compared them on our data. So it was almost, like we were pretending, well, what if we only had 2 hours of data? What sort of estimate would we get? And what we found was that all of the models provided a good fit over the duration for which the data were specified. But once we started making assumptions about after the data, we no longer were getting good fits to the data. So you can sort of see that here in the 2-hour exponential, we have the good fit, but then after 2 hours, we assume this exponential decay in the rate of appearance, which translates to this overall decay in glucose that doesn't really match the time scale of what we're seeing in the data. And if we see this in another participant, you can see sort of more extreme. And the fact that we have multiple insulin peaks which reflects some amount of insufficient insulin release early on, is quite common in this cohort, but is something that that can be accounted for in the model. So we can get good fits over that range, but it's something that complicates the interpretation of what insulin action is actually doing here. And just as another note here, the estimated rate of appearance for glucose actually varies with these different protocol durations. And that, so, here you can explicitly see after the 2 hours, the exponential decay that we're assuming. And that's something that isn't very physiological, right? We would expect that this rate of appearance should be similar and that the estimates for this based on the data should be similar, because the data over the first few hours is not changing among any of these models. So when we looked at the estimated insulin sensitivities of these with the 6-hour OMM, we found that they were highly correlated. So really across the board, everything was highly correlated, but some of the estimates based on some of these models were biased, and there also was more variability in some of these estimates. And so when we thought about, the overall research goal here, we wanted to detect impaired glycemic control in this population of girls with polycystic ovary syndrome. And so this is showing you what the difference in insulin sensitivity of these was when we looked at the PCOS versus the obese control on the 6th hour, where we actually had the full data set coming back to baseline. And with the limits of agreement that we sort of identified here, we determined that the 4-hour truncated model, so not making any sort of exponential assumption, would be able to differentiate the insulin sensitivity between these groups. And so, so this was great. It's still a long time for an ODTT, but it does cut 2 hours off, and that we're still able to get good estimates. It raises some questions about trying to compare these things across different data sets that I think are very applicable to a lot of the kinds of approaches that people use in informatics. So I think there's some great potential for more work in that space. So essentially here we just demonstrated that these estimates do depend on model duration and assumptions. And here overall, we were able to identify a model that performed well relative to the full model, but didn't take quite as long. The model also accounts for these complicated patterns of insulin release, but it's sort of a non-physiological assumption that pushes it into the rate of appearance. So that's something, again, to consider when working with a population like this. And so some of the next steps that we have with this work is to establish how these specific features of glucose and insulin profiles impact the estimates of insulin secretion. We've previously done work thinking about this idea of a single peak versus multiple peaks of insulin, and but it's not really known how that impacts these models in this way. Other features like reactive hypoglycemia, which is also very common in this population. And we also have done, so Melanie has run some very involved stable isotope tracer experiments with this so that we can actually pull apart the tissue specific insulin resistance here. And so working on modeling that data is also on our list with those data. Okay, so just quickly, I just have a quick time check.
11:27.
Thank you. Okay, so just want to make sure that I get through all of these things. So I wanted to talk about this project on modeling glycerol insulin dynamics, partially because it shows some of the opportunities for creativity that we have in modeling some of these metabolites that haven't been modeled before. So our motivation was the idea that glycerol dynamics during the oral glucose tolerance test could provide insight into insulin resistance within the adipose tissue. And there have been some previous models that were used to describe the dynamics of free fatty acids. So we thought that would be similar, but this hadn't been seen or hadn't been developed for glycerol previously. And so, as I mentioned previously, there was some insulin resistance in adipose tissue that had been identified in the girls with PCOS using the clamp technique, but the clamp doesn't really address dynamics. So it shows concentration dependence. And so when you use these different phases of insulin, you can assess insulin resistance sort of in different tissues, or the interpretation of it as insulin resistance in different tissues, but it doesn't address the dynamics in which that's happening. And the work that had previously happened with free fatty acids suggested that there wasn't really a dynamics issue to worry about. So we went into it not expecting to find a dynamics issue to worry about, but we did find one. So the schematic for the glycerol model that we ended up developing, same sort of idea of this compartment of the glycerol that we're measuring in the plasma. And here we're accounting for insulin independent and insulin dependent lipolysis. So instead of having insulin acting on the uptake the way that it was in glucose, we have insulin acting on the production, and where it's coming from lipolysis. The same sort of idea for insulin action. So you can see the equation that we ended up using for insulin action is exactly the same as it was in the glucose model. But here we have the glycerol model, where now the uptake of glycerol is just proportional to the glycerol concentration, but the production involves both this insulin independent and insulin dependent term. And this shows you the overall data that we have for this population. And so you can see, again, the same glucose and insulin type profiles. And now glycerol, as we would expect, as that insulin starts to rise, glycerol concentrations fall as lipolysis is suppressed. It's not fully suppressed, so we have glycerol concentrations kind of hanging out down here while insulin and glucose are high, and then we have this recovery. And notice that even in the average here, we have a recovery that comes up above baseline. And so some of the challenges that we had in thinking about this, so first, when we tried to use, we had the same insulin equation, so we just tried to use one of those and do glucose, glycerol, and insulin, and we could not get a good fit to the model. So there wasn't a parameter set that could do this. And as we started to dig into where this issue was coming from, it really had to do with the dynamics of the insulin action. Another challenge in this was that the structure of the glycerol model couldn't account for the glycerol concentrations to increase above baseline. So essentially they were maxed out when the insulin concentration was low, the insulin action was 0. And so we didn't have a way to account for that sort of overshoot. So when we thought about this, we decided to separately model insulin action for glucose and glycerol, and then compare the dynamics to see what was happening with this. And we also chose to truncate the glycerol data to fit at the glucose concentration nadir, and then minimize, by doing that, to minimize the time that glycerol was above baseline. So one of the reasons that we chose to actually truncate the data in this case, instead of considering the whole profile, was because the physiology was changing, right? So when people were going up above baseline, this was typically a counter-regulatory response to the fact that they had experienced hypoglycemia. So in the glucose falling, it actually fell below baseline levels. And then there was this response to bring it back up, which stimulated lipolysis at higher levels. So because it was different physiology, it did not make sense to try to represent that with the same constant parameters in the model, right? And so we chose to truncate that so that we were basically looking at at a period with more uniform physiology. So here's an example of a fit here in the representative participants, and you can see where that is truncated. And so we get nice fits to the glucose data here and the glycerol data suppressed in red on this axis. I'm just showing the insulin concentration so you have a reference of what the insulin is doing during this time. And then from this, this shows then what the modeled insulin action is doing. So remember, the insulin action is sort of a transformed version of what the raw insulin is doing. And we tried to identify some various metrics that could be useful for determining what differences we were seeing in the insulin action here. So we thought, we can look at differences in the peak timing of insulin action and how that relates to the peak timing of insulin, how that relates between the peak timing of insulin action on glucose and glycerol. We can also see where the insulin is when the glucose is at its nadir. So remember, glucose is already back down. If the participant is experiencing reactive hypoglycemia, it may even be lower than baseline. And if we're in a case like this, where the insulin action is still really high, then that's something that's going to be continuing to push the person down, right? So we wanted to understand what was happening there. And this basically just shows that in all of these measures, we found that the glucose insulin action was delayed relative to insulin, relative to what was happening with glycerol. We won't go through those in detail, but this was also true for the dynamics. So this parameter P2 was specifically governing the dynamics of this, and we had an order of magnitude difference in the sort of distribution that we had across participants that we can see here for the glucose versus the glycerol. We can sort of visualize this, thinking about this as a phase plot, where we plot the normalized concentration of the metabolite against the normalized insulin action. And so we get this cyclic profile for glucose and glycerol, where an increase in glucose here is followed by an increase in insulin action. Whereas for glycerol, and so then we get this cycle. But for glycerol, we really have much more of this sort of straight out and back because it's tracking so closely. So some quick conclusions here was that we were able to fit the data really well using this model and by restricting the time interval for when we were doing this. And using this approach, we really identified this difference in the kinetics of what was going on, so something that hadn't been reported previously. So really, these are preliminary results. We've only seen them so far in this one cohort, and this is a cohort that has high insulin resistance, but we're very interested in sort of diving into this deeper to really understand what's going on here, and if this sort of mismatch of dynamics is really a hallmark of the insulin resistance that we're seeing in this population. And then from the modeling, on the modeling side, we're also really interested in extending the model to eventually account for a greater range of dynamics, as happens in the reactive hypoglycemia. Okay, so then my third project that I wanted to talk about is modeling C-peptide to assess beta cell function in patients with cystic fibrosis. And this is work with Dr. Christine Chan and my current PhD student, Justin Garish, and one of my colleagues in statistics at Colorado School of Mines, Doug Michka. And so when beta cell function is reduced, particularly in the early stages, there can be insufficient insulin release, particularly early on in the insulin response. C-peptide is co-released with insulin and gives us a reliable marker of insulin secretion. Insulin itself sort of involves insulin secretion and disposal. And beta cell function is something that we know is impacted by disease, right? So this is a primary issue in type 1 diabetes, can happen in type 2 diabetes as well. And then this happens in cystic fibrosis with cystic fibrosis related diabetes. So most of the sensitive indices that people have used for quantifying beta cell function rely on reliably estimating insulin secretion rate. And insulin secretion rate is one of these things that we can't measure, and so we have to infer it somehow. So why are we doing this in a population with cystic fibrosis? Well, cystic fibrosis, to remind you, is this genetic disorder that causes mucus to build up in small ducts and can severely limit life expectancy. And one of the organs that's impacted by cystic fibrosis is the pancreas. And so the disease can directly lead to the damage of beta cells, which affects insulin secretion and may worsen over time. So cystic fibrosis related diabetes can follow this and is the most common comorbidity that's associated with cystic fibrosis. So we wanted to try to infer insulin secretion and use that to quantify beta cell dysfunction in a cohort with cystic fibrosis. But there's a lot of uncertainty in using these models and this approach in people with cystic fibrosis. So clearly none of the validation or anything else has happened there. We are going to infer the insulin secretion rate from C-peptide data. As I mentioned, C-peptide data is co-released with insulin, but it's a more reliable marker when you want to focus on insulin secretion. So because of this uncertainty, we decided to use a hybrid modeling kind of approach. And I think, hybrid models are generating a lot of excitement and really offer the best of both worlds in a lot of ways to some of the more first principles based models that we sort of have traditionally used, differential equations and things that kind of encapsulate what we know about the system, as well as database models that account for what we're seeing in the data, but maybe we don't understand the mechanism that's directly representing them. And so to do this, starting off with our first principles based model, we have this idea of C-peptide, where this is the C-peptide that we're measuring. There's some clearance of that. There's some exchange between intravascular and extravascular C-peptide compartments. And C-peptide overall is driven by this underlying insulin secretion rate. And so the model equations for this were first proposed in 1980 by Eaton and colleagues. And so this sort of shows you what I was showing in the schematic, with the key focus being on this unknown forcing function of insulin secretion rate, S of t. And so the C-peptide data that we're working with here, this just shows you an example of what's happening with the glucose and insulin, sort of following the same profile that we've been talking about. And then C-peptide, as you can see, sort of following along with a similar profile to insulin. And this was, again, a relatively frequently sampled OGTT over 3 hours. So as I mentioned, some of the challenges in our C-peptide data is that there's a lot of uncertainty in this cohort, and we want a convenient way to quantify the uncertainty in our ISR estimates. And so, basically, we wanted to use a structure that not only did we have a stochastic component in our hybrid model, but it was a stochastic component where we could quantify uncertainty and then propagate that through to other metrics that we wanted to derive. So this sets up the Bayesian hierarchical structure of our data process priors levels. And at the data level, that's coming from the ODE model. The MN is just multivariate normal, where we're drawing these. And then because our C-peptide system is linear, except for that insulin secretion rate, we can use an integral equation transformation on this to represent our C-peptide above basal. And we assume this mixed effects model for our insulin secretion rate, where we just had a fixed part, and we just chose it to be linear as sort of a first guess, and then a stochastic part that really sort of accounts for what's really going on here and is the realization of a zero mean Gaussian process. So we didn't use a purely Bayesian approach here. So we derived the parameters for our statistical model empirically. And although we had relatively sparse sampling in our data for individuals, we had independence among participants. So we were able to draw strength from that in our maximum likelihood function. So this just shows you the estimates that we got for population levels for those parameters. And then again, since we can't measure insulin secretion rate, this was something that we wanted to validate on synthetic data so that we actually knew what we were starting with, and then we had a good idea of how well we were actually reconstructing that. So this is a simulated insulin secretion rate that I'm going to take as my true ISR, and then feed that through the differential equations model to say, what is the C-peptide that's going to be produced? So you can see the exact values of C-peptide that would be predicted by this model in the blue dots. And then we added some noise to perturb that to represent what would happen if you were just, sampling and you have some error in your measurements. And then we used that observed C-peptide as our data and used this approach to reconstruct our insulin secretion rate. And so you can see here that our exact insulin secretion rate that we started with is in the red dashed line, and then the inferred insulin secretion rate is in black. And so we did a really good job and had, not only a nice fit sort of overall, but had this within the intervals where we were expecting this to be predicted, the credible intervals. So then if we take that insulin secretion rate and go back to predict C-peptide, then again, we get something that does a good fit through that C-peptide data. So that just kind of closes the loop of what we were doing. And so then we turned to the clinical data. And so this shows 4 representative participants who have very different C-peptide profiles. So you can see their C-peptide profiles. The data are these dots that are a little bit small here, but the red dots. And then you can see the inferred insulin secretion rate for each of these people, and then the predicted profile that comes out of that. And so although we had a lot of different secretion rates reflecting different stages of pancreatic function here, we actually were able to describe all of these with this approach. So we had that flexibility. So remember, when we started off, we wanted to take that uncertainty and be able to propagate it through into these measures. So the kinds of measures that people would use to think about beta cell function or pancreatic function based on insulin secretion rate are looking at the 3-hour insulin secretion rate, so that and the area under the curve there. And so this shows the 3-hour area under the curve for my 3 different groups: the control, the CF, and the cystic fibrosis related diabetes group. And here we have all of these with confidence intervals, and our total 3-hour insulin secretion was shown to be increased in both of the cystic fibrosis populations relative to the control. But if we look at the acute insulin secretion rate, so what is the insulin that's released in that first 30 minutes, then we see that that is something that's higher in the control group compared to the cystic fibrosis groups, right? And both of the cystic fibrosis groups. And so this is showing that even before the progression to cystic fibrosis related diabetes, we have this reduction in beta cell function, especially in that initial response that eventually requires more of response later on and leads to more insulin being secreted overall. So this just sort of wraps up what I was saying about the C-peptide modeling. And we're working to refine this model and apply it to some exciting new data that Christine Chen has where we want to really sensitively look at this question. So I realize I'm just about out of time here, so I'll just very quickly touch on this project where we use these kinds of measures to think about relationships with sleep and circadian rhythms. So this was in adolescent girls with overweight, obesity. Some of them had polycystic ovary syndrome, and we used actigraphy to assess sleep timing and salivary melatonin to assess circadian phase. I won't go into the details about what we all of the things that we found here, but we found that short sleep duration was associated with reduced sensitivity. Also, late bedtime or chronotype and late circadian timing were associated with reduced sensitivity. So this is kind of similar to the findings that have been reported in adults, and we saw that come through in our population as well. Has some significance, particularly, as I mentioned, for adolescents who already have later circadian timing. But one of the really interesting findings in this study was that we actually identified that polycystic ovary syndrome status is associated with morning circadian misalignment. And so this was a really interesting finding, sort of relating this metabolic profile to something that's happening on the circadian side. and really unpacking this and understanding where this might be coming from, and if this might be a realistic therapeutic avenue or a place for interventions is an active area of research right now. So, all of these models and methods that I talked about today are things that can provide some good insight into what's happening, but there are lots of opportunities to extend this and think about it in the context of other data, especially when we're thinking about participants with diverse metabolic phenotypes. These are models that are typically developed in sort of healthy adult populations, and so being able to apply them broadly and really, use their potential for realizing some of the personalized medicine goals that we all have requires adapting them. Obviously, we're not going to be validating them in every individual population, so we need reliable ways to adapt these and make sure that they're being used appropriately in a range of populations. And particularly thinking about these sleep, circadian, and metabolic interactions, I think is exciting because it gives us some great opportunities for targets for intervention that can improve health. And really, I think sleep and circadian interventions are so powerful and yet also so simple in some ways, right? If you can actually get people to sleep a little bit more and maybe go to bed earlier and have that have a big impact on metabolism, that's really wonderful. I didn't have time to talk about my sleep and circadian modeling, but again, I'm happy to talk about that. And this just sort of flashes up, interested in questions in development, interested in sleep disorders, and circadian things. These are just some of my funded projects and pending grants. And so I'll acknowledge my students, my collaborators, and I'm happy to take any questions.
Do they put questions in the chat? I can or raise their hand. I can unmute them. I'm not seeing anyone.
So I guess one of the questions that I had is thinking about like CF related diabetes. Is the mechanism of beta cell dysfunction known? Like how CFTR changes beta cell dysfunction?
Yes. So within the pancreas, the sort of same problem is happening with the duct. And so this causes some of the digestive things that are being released by the pancreas to back up into the pancreas and cause it to sort of destroy itself. So it results in inflammation and scarring that reduces pancreatic function.
And then there's new therapies that prevent CFTR recycling in the context of like a delta F508 mutation, right? And so do you have, like it seems to me that the introduction of those therapies for patients could give you like a moment in time where that behavior changes. And do any of the data sets that you have kind of span the introduction of those therapies? And does that give you an avenue to kind of understand if this process is likely, if those therapies or therapies like them are likely to halt this process, or if it's going to continue to be a challenge going forward?
Yes, that is an excellent question. And that's exactly the exciting data set that I mentioned. So we have before and after Trikafta. And so one of the things about that is it seems to be showing great promise in increasing life expectancy, which is wonderful, but also could be associated with a big increase in CF-related diabetes as people are living longer. And the effects of it on metabolism are not really well known. And so this is actually an active project, so stay tuned. But our preliminary results show that there is a direct effect on metabolism. And actually, it's very exciting because it's not clear, because there is this damage to the pancreas. It's not clear that it would be fixed, but it seems that reducing the inflammation and all can actually improve that. So yeah.
Here's a question online from Christine Chan.
Oh, all right.
Do you want to read it? Or-
Yeah, hi.
go
So
ahead.
I was just gonna, can you hear me? So,
Yes.
okay. Yeah, I was just gonna add to that comment and regarding CF, CFRD pathophysiology. It's, I think there's a lot that we've learned in the last few years, 5 years even, about the pathophysiology of disease, and still a lot to learn. Increasingly, studies have shown that yes, beta cell mass is decreased, but that alone doesn't seem to be enough to explain the progression and development of CFRD. And there's been a lot of nice work. Dr. Rebecca Hall from Seattle came and spoke at the BDC not too long ago, investigating the pancreatic milieu. And increasingly, we're learning that there's likely not just there's inflammation, systemic likely and local, affecting beta cell function, but increasingly there are likely interactions again between the milieu, the ductal cells, and also the intravascular and likely neural networks, and paracrine signaling affecting beta cell function. So the effects of CFTR modulators on the disease course of CFRD still I think remain to be seen. I would say, there was a lot of controversy and interest in whether or not CFTR exists in the beta cell, and the prevailing, this is a whole talk in and of itself, but the prevailing data seem to suggest that there are not significant amounts of CFTR in the beta cell. So, if we're going to see any change in the progression or pathogenesis of CFRD, it'll likely be from, indirect effects, I suspect. And again, there are limited, really small studies looking at pre-post modulator effects on insulin secretion, and really pretty varying data. And I would say that at least in the short term, there don't seem to be, huge changes in terms of insulin secretion from CFTR modulators in terms of making the diabetes go away. So we'll see. And we do have a data set where we have frequent sampled OGTTs, pre and post, roughly about 1 year post modulator. And Cecilia is helping us to look at that. So more to come.
Thanks, Christine. Yeah.
Yes.
So I was wondering, in the different sort of situations you've been looking at, have you thought about how you could account for, say, things like glucagon changes and glucagon regulation or glucagon action, or could that be sort of separated from insulin sensitivity, or does it sort of fall in the same sort of?
Yeah, no, that's a great question, and it's definitely something we've been thinking about, particularly in the context of thinking about the counter-regulatory response. It's not something that people have generally included in these types of models, although there was a model published last year that did include glucagon. So I think, the field is definitely going there. Yes.
This question is about the approach. What do you think about approaches like constraint-based modeling, like flux balance analysis, which of course doesn't have this dynamical properties, parameters and all that stuff, but could give you this opportunity to incorporate tissue specificity and even genetics data overlaid on top of these enzymes.
So sorry, can you tell me more about what kind of approach you're talking about?
The constraint-based modeling is where you don't have any equations, but you look at the stoichiometry matrix and then assume steady state, and then do quadratic programming or something to figure out the concentrations or the fluxes through every single equation or metabolite or reaction that you have in your model. So that allows you to then say, okay, I'm going to overlay gene expression data on top of it, or genetics on top of it, and say, I'm going to try to make this model specific to this particular tissue by adjusting the concentrates of the enzymes proportional to the expression level of the genes that encode them.
So, I think there's a lot of space here for lots of different kinds of modeling approaches. So, it sounds like that's very promising. I think, it is important to think about what these things are doing at different stages. Because, for example, if you just look at basal levels of insulin and glucose, you can get a lot of information there, but you don't know what the maximal insulin release is going to be, right? And so somebody could have the same basal level and have a lot of insulin hypersecretion and go really high, leading to a very significant glucose crash, and somebody else with that same basal level could have a more typical insulin response. So really looking across that time point, you could get lots of different information, but I think then layering that in with other kinds of approaches could be very powerful. Yeah.
Does a patient have the glucose tolerance test? Because do we have both males and females?
Do we have
Males and females?
Yes. So a lot of the data that I was showing today was in females, but we do have some data in males as well.
Okay. And then do you see differences, especially if you look at the adolescent?
Excellent question. So yes, there do seem to be potentially sex differences, and understanding that in more detail is also a very active area of work right now. So yeah, I'll talk about that more tomorrow too, because since that's a proposal that we want to do soon.
All right, thanks.
We're good. There's no more questions. I think we're at the time, so.
All right.
Thank you.
Okay.
Thank you so much.
Thanks.